Hi, I'm Sarah Light, and I'm here with the Soil Health Connection. I'm here with Liz Harper, Executive Director of the Colusa Resource Conservation District, and today we're interviewing Dr. Tim Bowles. Tim is an agroecologist in the Department of Environmental Science, Policy, and Management at the University of California, Berkeley. Welcome to the show, Tim. Thanks for being on.
Yeah, thanks for having me.
Your research has focused on a very important connection between soil health and soil fertility. As an agronomist, I find that to be particularly interesting. And certainly from a practical perspective, it's really important to understand how the two intersect in order to make on-farm management decisions. Specifically in your work, you have found that we may need to rethink crop nitrogen availability for systems with high soil organic matter. How should we think about the pool of available soil nitrogen?
Yeah, that's a really good question. And I would encourage us to think about this as both a conceptual and a practical shift in our thinking. I think first, thinking about a conceptual shift. We talk about this pool of available nitrogen, but a pool seems kind of static, right? And instead, I want us to think about it like this really dynamic system. So I think about it like, if we think about it like a pool, it's like a pond or a lake or something, right? And there's just a certain amount in there, but we don't think of it as having both like inflows and outflows. So instead, I like to think of it as like a nice little wading pool that you might find, somewhere along the Yuba River or something like that, right? So you can go for a dip. It's got a nice pool there, but it's got an inflow and an outflow. And that's what's happening in the soil. We think about the forms of nitrogen that are most available to plants, ammonium and nitrate, right? And we can measure, that's what we measure when we do a soil test, right? We measure the amount of ammonium and nitrate that's in the soil. A lot of times we just focus on nitrate. But when we're doing that, all we're measuring is like the amount of using this sort of metaphor, we're just measuring like the amount of water that's in this little wading pool, but we're ignoring the flow that's coming in and the flow that's going out. So the flow that's coming in is all of the activity of microbes. It's all of the decomposition that's happening, that's transforming it's transforming organic forms of nitrogen from the soil or from organic amendments into these inorganic forms of nitrogen. And then the flows that are coming out of it is everything that the microbes are also taking up and assimilating and making part of their own bodies, but it's also the nitrogen that's being taken up by the plant roots. So we could think about what that means practically speaking, right? If we think about this conceptual shift from thinking about it from just a pool, a static pool, to this really dynamic system. Well, that means that we might measure a relatively small pool, but if the inflow and the outflow are really high, well, that might not mean that there's actually nitrogen limitation happening. It might not mean that plants are actually limited by the amount of nitrogen that's in the soil because the rate of nitrogen being produced and taken up is really high. On the other hand, we might measure a big pool, but there's just a trickle coming in and a trickle coming out. So that pool may just be kind of sitting around, and as it's used up, there may not be much more coming in. So there's this disconnect between how we measure nitrogen availability in the soil by measuring these pools and what's really happening, which is super dynamic. And that becomes even more dynamic as we build soil health, right? Because we think of a healthy soil as having a number of different characteristics. Oftentimes soils with higher levels of organic matter that fuel more microbial activity. As organic matter increases and the activity of those microbes increase, that those processes that release inorganic forms of nitrogen from organic nitrogen in the soil accelerate. So oftentimes we see these flows increasing, right? And so that leads to maybe shifts in how we think about what we're measuring here, right? And how we interpret those measurements. That is, if we measure a small pool of nitrate or ammonium, well, does that actually mean that there's little nitrogen that's available for plants? Or could it mean that the flows are high, but just the amount that's building up at any one time is not very high? So that's, I think, the that conceptual shift that, particularly as we're building soil health, we need to make, and particularly as we're kind of thinking about sources of nitrogen fertility other than synthetic fertilizers, right, that are supplying those inorganic forms just directly, no microbial processing required, right? So in those circumstances, when we're building soils that are healthy with a lot of microbial activity, a lot of available organic matter for those microbes to chew on, and when we're not using synthetic sources of fertility, then that's when we want to kind of start to make this conceptual shift.
Excellent. So optimizing nitrogen management is important and a recommended best management practice for many crops. Is soil sampling in the root zone prior to in-season nitrogen application. This practice is more common in conventional systems, but what recommendations do you have for soil sampling methods and frequency for growers with high soil organic matter?
Yeah, that's a really good question. And so I'll kind of build on both these conceptual shifts that I was mentioning, but also some research that I've done in the past in the Yolo County area on kind of working organic farms. The first thing I'll say is that the work that I've done with others really is bringing some nuance to a really tried and true set of principles that exist for managing nitrogen, both in conventional and organic farms. And so I think we're going to link to a new resource on how assessing nitrogen availability, particularly for organic farms, but the principles apply to anybody using a variety of fertility sources, especially organic amendments. And yeah, in that framework, assessing kind of in-season or just pre-season nitrogen availability is part of this whole this important framework, so that you can adjust kind of in-season fertilizer application, whether that's synthetic or whether it's applying fish emulsion through a drip line or an extra side dressing of guano, or anything that's applicable to any of these systems. And what we've found on kind of working across about a dozen organic farms growing mainly summer vegetables, we were focused on tomatoes, is that as on the farms that had higher levels of soil organic matter, we're talking about 2% or so soil carbon, which translates into a little over 4% soil organic matter. As we kind of reached those higher levels, which is fairly high for the Sacramento Valley area, what we were seeing is that we were measuring very, very low amounts of nitrate and ammonium in the soil. So the pool size of nitrate and ammonium was really low at any one time. This was measured both right before tomatoes were transplanted and then around tomato flowering, which is right before this kind of exponential growth phase begins. And that's when the plants are really trying to take up most of their nitrogen, right? And that's the time, that time period is when you want to focus these soil measurements. But we found this disconnect, right? Because we were measuring really low amounts of nitrate and ammonium in the soil, but the plants showed, then this was below the threshold for what should have been, should have required additional fertility. But when we were looking to the plants, we weren't seeing that same evidence. We could do tissue samples or whole plant samples, and they showed their nitrogen levels were just fine. The yields were on these farms were at or above county averages. And so when we paired and we started looking at a broader set of data, we could look at other measurements, more specialized measurements that were looking at the amount of like active organic matter that was there, the amount of microbial biomass that was there. And on these farms that had really low levels of ammonium and nitrate in the soil, we found really high levels of active organic matter, of microbial biomass, indicating that what was happening was kind of like I was describing in this kind of, in our wading pool example, right? That the actual pool of nitrate and ammonium wasn't really building up, but there appeared to be a really high rate of nitrogen becoming available through the activity of microbes, right? And so for those growers, I mean, one of the growers that had been, doing organic for like 30 years said, look, we've always seen this, or for many years, we've seen this disconnect between these nitrate tests that we do and what we just actually observe in the plants. And so we just ignore them. We don't pay much attention to our nitrate tests anymore. And this, I think, helped explain a little bit of this puzzle. And so the idea is not that assessing soil nitrate availability or other during preseason or pre-side dress isn't useful. It's just that we want to think about these kind of sufficiency thresholds and the extent to which they apply or don't apply when we're building up really active soils. And how we might be able to use a little bit more information, either past experience or other lab measurements, to help contextualize those nitrate measurements when we do find them low. Because you want to know, is this a situation where we have nitrogen deficiency, or is the system really cranking kind of like it's supposed to?
Yeah. So you never really kind of mentioned that the plants were taking nitrogen outside of ammonium and nitrate in these lower nitrate systems, or in the systems with higher organic matter. You just kind of referenced the microbial activity. So does this mean that the plants are taking up other forms of nitrogen?
We didn't assess that directly. And we do know that it is possible for plants to take up other forms of nitrogen, particularly soluble organic forms of nitrogen, amino acids, for instance, right? Proteins are made of amino acids. And the first step in the decomposition in soil for breaking down these large organic molecules is breaking them down into these individual components, things like amino acids. And actually, the concentration and the cycle of amino acids in soil is also quite dynamic. And we know that certain plants can certainly have the ability to take these up. They have the transporters that can grab these out of soil and take them into their roots. What we don't know is how quantitatively important this is, particularly in agricultural systems. Historically, it's been thought that it's really not important. And we also know that since most of our crops, or many of them, are evolved from more ruderal type species, more weedy species, those species tend not to rely much on these other forms of nitrogen, these more soluble organic forms of nitrogen. The plants that have evolved to rely more on those tend to exist in really low nitrogen environments, and where the competition for nitrogen is very high. And so we didn't measure whether other forms of nitrogen were being taken up, but it is possible. I think and it is something we should look into. But just based on kind of what we know about the evolutionary history of most of our crops and what we might expect, is that it's probably not the most important pathway for nitrogen uptake, even in these systems.
So the second part of that question was, if you were to give advice to growers of how to optimize their nitrogen applications in both organic and conventional systems, what would be the one thing you say? It could be many things, but.
Well, I will point. I want to point to this new A&R publication from Margaret Lloyd and Daniel Geisler and others, which I think really synthesizes a lot of work and thinking holistically about nitrogen management. And then I think I would repeat a couple of pieces of advice. Well, repeat one piece of advice that I think I always lingers from one, a conversation with one of the growers involved with this study in Yolo County that I mentioned, which was, pay attention to the plants. We can look at what's going on in the soil to help give us clues, but at the end of the day, pay attention to the plants. And what I would add to that is that, we're looking, I think, ideally for optimization here, right? We, know that we have to produce healthy, productive crops, and that, water quality and greenhouse gas emissions are a big downside of a fertility program that's not really well optimized. And so we can look to the plants, but then we also have to look at some gut checks about how well the program is working. And so that's where I think not only kind of in-season soil measurements of nitrate and other more active organic matter measurements can be really helpful, but also looking at harvest, looking post-season, what's left after the crop is done. That can give you an indication of how much residual nitrogen you might be worried there might be, and what might be susceptible to loss, and if there could be opportunities for kind of decreasing that what's left over at the end in the future, so that we can arrive at, I think, this more of this optimization situation.
Tim, thanks so much for sharing that really exciting work with us. It's really timely for our state, and I know that our audience is really going to appreciate this interview a lot. Before we wrap up, can you tell us a little bit about what you're working on now and where people can find information about your projects if they're so interested?
Sure. Yeah, we have an active or somewhat active Twitter account at Bulls Lab. And we try to at least post a lot of nice pictures from our field work and some videos. And then I'll ask Liz and Sarah to share the link to our lab website, where we're working on getting our research pages up to date. A lot of the work that we're doing now, our main projects are down actually in the Central Coast, working with a wide range of organic lettuce producers, trying to understand the many factors that go into their decision-making around whether to use or not to use kind of soil health building practices, what the challenges and barriers are, what the innovations that they've come up with. So it's, and then how that affects some of the things that we're talking about here. So how it affects soil nitrogen cycling, soil water dynamics in ways that might reduce needs for irrigation, how it sequesters carbon in soil. So it's a big interdisciplinary project, and we hope to have some results out here in the next year or two.
Awesome. Thanks so much for being on our show today.
All right. Thanks so much for having me.